and knowledge in the field of towards next generation healthcare technologies. So, Dr. Pandya, over to you, sir.
Thank you, Palash. Yageshwar, Sivaraman, for inviting me for this talk. I'm really glad to participate today. I'll be talking about 1 hour, 45 minutes or 50 minutes. In between, I'll stop after a few topics to see if the participants have any question, and I'll try to answer with best of my abilities. Uh, I hope you can hear me clearly. One of you can just confirm, and then I'll share my the screen also. Palash, you can hear me?
Dr. Hardik. Yeah, you are audible, Dr. Hardik.
Yeah.
Thank you. Thank you. So I'll just stop my video and then I'll share the screen.
Yeah, sure, sir. Sure, sir.
Okay. Are you able to see the screen?
Yes, sir. Yes, sir. It's visible, sir.
Okay, great. So yeah, I'll be talking about next generation healthcare technologies using microengineered devices and systems. This falls under the bigger umbrella of biomedical and biomechanical engineering. It falls under bigger umbrella of MEMS-based sensors, chips, implantable devices, as well as sensors and transducers. As already Palash told, my name is Hardik Pandya. It's very easy to remember. And I am representing Indian Institute of Science today. I have been affiliated with some other universities as well as an associate faculty. But in the capacity of a professor here at DESE, I'll be talking on this particular topic. So, role of medical devices in healthcare. We'll start with that to see that why we are talking about these biomedical devices or biomedical technologies and how it will help us to solve the problems that are existing at present. So you start with a patient when he or she will feel a particular problem, right? We start with screening. So screening, when we talk about, falls under several domains. The first domain in screening is understanding the vitals. And now we all know due to COVID that understanding vitals are important to develop a screening tool. Few vitals that we already know are temperature, SpO2, right? Then we also know about heartbeats, right? We use a lot of Fitbits and other watches that we use for our health. So another area on this is a mobile stethoscope. If you have heard about stethoscope, there is another field called mobile stethoscope. And this all falls under the screening tool. Now, if you talk about cancer, particularly oral cancer or FNAC, then it goes to understand the morphology of the cells. When you want to understand the morphology of cells, it is called cytology studies. So the cytology studies comes under screening. Now, when a person is screened, right, and the outcome is suspicious, the person is asked to go for the diagnosis. So diagnosis, if you talk about, let's say, cancer, in cancer, the diagnosis is histology studies. Histology studies is study of tissues. So there are several biomarkers. We'll be talking about few biomarkers in the area of breast cancer. But in general, diagnosis is the gold standard. And based on the diagnosis, the treatment is further given to a particular subject. Now, so after treatment, there is a restoration period, right? And finally, when the patient is sent home, there requires a regular monitoring. So if you see, in general, the play of medical devices across the continuum increasingly leads to improved outcomes, it's healthcare outcomes, because if you have a tool like mobile stethoscope, you can even check your heartbeat at home and the data would be available at a remote with a remote clinician, right? So is diagnosis. What are the difficulties or limitations in diagnosis tool? And can we develop a tool that can be used or for the pathologist or can help the pathologist to make the diagnosis better, that is accurate? and rapid as well. So we always talk about developing a system which is affordable. Affordability comes at the last. First is always the accuracy, then how fast it is, and then comes affordable, right? We can always make things affordable, but we should not be compromising on accuracy or the sensitivity, specificity. This all falls under the accuracy domain. So going forward, if you develop a medical device, then when you talk about medical device, it has several interdisciplinary area, and that's why if you want to work in healthcare technologies, it's very important to work with researchers from different field, included but not limited to electronic systems engineers, right? Mechanical engineers, computer scientists. We have biologists, and specifically you need to collaborate with a clinician, right? Because you want to use human samples. It can be tissue, it can be blood, any sample. You sometimes, if you are, when you want to work on human subjects, then you need to, for example, if you are looking at the EEG data, right? I will talking about all these things. You require ethical clearance to get these subjects to be utilized for the technologies that you have developed. So very important to have a right clinicians on board working with you along with the other researchers that I already talked about. So if we work on improving the medical devices, then it will not only help us in better outcomes, healthcare outcomes, it will also reduce the length of hospital stay. Now, we all know that hospital stay, even though it is cheaper than most of the countries around the world, right, it is still expensive based on the income of a family. So if you reduce the stay at hospital by developing a monitoring tool that can be used at home, right, you can save a lot of money. Next one is, now this saving of money is different than developing an affordable device. Okay, so we should always look at the reducing the out of pocket expenditure of a given subject or a given patient. Accurate diagnosis and targeted treatment. Now, you see, I'll give you an example. I don't know whether I'll have time to talk about it, but if we developed, if a person has, let's say, a certain disease, let us take example of cancer again, and if there are 3 drugs available, all 3 drugs are meant to kill that particular cancer. Which drug would be more effective for that particular patient? We don't know, right? So the best way of working right now, when I talk with my friends who are clinicians, is that based on experience or based on the last patient who was suffering from the same disease, they administer the drug. So the point is, can we develop a patient-centric platform by mimicking the cancer, by taking the cells from the patient, developing in the laboratory, and using those drugs and tell which drug would be more effective? Effective drug means its efficacy is very high. So can you develop a patient-centric platform to understand which drug would be having a higher efficacy, and that drug you can administer to a patient? That comes under a patient-centric platform. Targeted treatments. If you understand chemotherapy, which all of us know, right? It has a lot of limitations. But if you understand immunotherapy, then the immune cells are activated by blocking some markers. For example, there are PD-1, PD-L1. PD-1 is on the T cells, which are white blood cells. PD-L1 will be on the cancer cells. If you block one of the marker, it will have more effectiveness towards treating that particular cancer. Immunotherapy, targeted treatment, that comes under targeted treatment. Next is, if you have this kind of advanced medical devices, then you can reduce the burden of medical resources, finally increase access to care. So if you, we always complain about the medical infrastructure, less number of doctors, but we should also understand the population, right? So of course, we should work on increasing the hospital beds per 1,000 people, but we are on that path of improving all the facets where we were lagging behind, in previous few decades. Right. So now we are on the right path. We are trying our best, working with each other, collaborating in a right approach and manner to increase all this or overcome the limitations that are in front of us. The first limitation is less number of beds. How we can increase? By having more number of medical hospitals, right? Medical centers. So 1.3 as of now, 0.7 doctors per 1,000 people. This should also increase. And that's why more number of medical colleges will lead to more number of doctors. And that's why this 0.7 ratio would change. 18% of the communicable disease. Communicable is where the diseases are transmitted. COVID is a communicable disease. But non-communicable diseases are like cancer, right? So these are non-communicable diseases. So higher penetration of medical device could improve the access to healthcare. Now, this slide is very important for us to know so that we understand that why we are working in healthcare, what is the importance of biomedical engineering, and how we can work in an interdisciplinary manner to develop advanced healthcare technologies. This is something that we need to work extensively. If you see, the Indian medical device industry presents just over 1.3% of global medical devices, where for the USD 335 billion, which are dominated by US, Germany, France, and Japan. So with the new policies coming in, with the ecosystem support, with the industry estimates, with the faculty development programs like this, right, we try to meet each other. We try to understand the collaboration efforts. We try to understand how we can work in a right manner to develop and work with industry to develop new medical devices and increase this 1.3% close to 28%, which industry estimates a potential grow by 2025. This is a optimistic value that we should target, but this will only happen when we work with each other, right? So if you see from 2008, 2014 to 2020, there is an increase in the ratio, in the percentage, right, of the consumable implants, diagnostic imaging instruments and appliances, patient aids, and other. Several verticals are there, which I talked about, like consumable implants, diagnostic imaging instruments, and patient aids. So what we'll be talking about in today's discussion will be on medical and surgical instruments, cancer diagnostic tools, point of care technologies, and healthcare and telemedicine. So this is very interesting plot, right? That if you see the total market size of the medical device equipment versus total local production, so that all things are doing really great. But when it comes to exports, right, we are not exporting that much, but we're importing a lot. So there is already a burden on the taxpayer to import, because all these imports are done through the government bodies, and government bodies are funded through the taxpayers. So a lot of burden is on the common man, because we are importing large number of medical devices rather than we should be an exporter of these devices. So that is the reason that I chose of using the electronic system understanding, sensor design understanding, understanding about a little bit about image processing, machine learning, right, about mechanical engineering to work towards healthcare technologies. So the key focus area in my lab is on biomedical devices, and some of the disease that we target is on breast cancer, oral cancer. We work on developing catheters for treating the heart disease like atrial fibrillation. We work on developing catheter to understand the tracheal inflammation. We work on developing a probe to delineate normal from tumor in case of brain cancer. We work on developing a band that can help to understand hearing screening. We work on developing a microscope for understanding the cytology-based studies in case of cancer. We also work on developing a biosensor or a point-of-care system for understanding the antibiotic susceptibility and protein-protein interaction. And finally, we are focusing on surgical tools as well. So in a way, we try to expand our understanding to develop technologies to solve some of the problems that are in front of us. Now, when we talk about technologies, we need to also understand the size at which these sensors are fabricated. We all know, if you have studied electronics, that the world is moving towards 3 nanometer technology, right? That means channel length is getting smaller and smaller. The reason being that the speed of the transistor would increase, and the number of transistors that you can pack in one particular silicon wafer would also increase. But when we talk about sensors, right? Sensors can be nanofabricated sensors, can be microfabricated sensors. So microfabricated sensors, actually sensors are of few millimeter, but the dimensions, the patterns that we develop are of few microns. And that's why what is one single micron and how large is one single micron? If you see, of course, we can say that it's 10 to the power minus 6 of meter, but that is not how the answer should be. We should also see in terms of some example, right? So this is an example. If we take a strand of a human hair, it would be about 50 to 70 microns, right? And of course, we have a little bit thicker hair from north side to south side when we go, right? In our country, we will see the change in the thickness of the hair. But it will fall between this 50 and 80, 50 and 90 microns, all right? Same way from west to east, you will see there is a difference in the thickness of hair. So the point is, one single strand of hair is about 50 to 70 microns. If you take a pollen, right, we know there is a lot of pollen energy. This single pollen is about 30 to 50 microns. Take about ragweed, it's about 17 to 23 microns. Take about dust mites, it's about 10 microns. So 1 micron, if you want to see compared to this, some of the examples, will look like a dot, as you can see in this particular slide, right? It looks like a dot. Let me just see if I can put the pointer. Yeah. So it's like this. It's smaller than this pointer, in fact. So that is our 1 micron. Now, what you're talking about is a few nanometer technology. So you understand the dimensions effect of all these things. Now, why we talked about microns is because the technologies or the chips that I will be showing it to you as a part of this talk will be of few microns. The patterns would be of few microns. Let us start with the first topic, the clinical challenges breast cancer. And we need to see why we had to work on developing a technology for improving the accuracy of the current standard, or what are the gaps in the current technologies, so that we can fill those gaps using the developed technology, by developing technology in the laboratory, and so on. So if you see this graph, first let us see the pie chart. In the pie chart, you will see that compared to all the cancers, right, breast cancer is about 11.6%. This is the incidence, but the mortality is about 6.6%. So that's one thing. Second thing, if you see from left side to right side in the bar graph, right, what you see is that the incidence rate in the countries like Australia, New Zealand, Western Europe, Northern Europe, North America, Southern Europe, Polynesia, right, is really, really high. But the mortality is really low. I'm sorry, this should be red color. So anyway, this mortality, this red bars are really low. As you go towards the right side, right, to countries like Central America, Southeastern Asia, Western Africa, Eastern Africa, Middle Africa, South Central Asia, including India, right, the incidence is comparatively low than these countries, but the mortality is very high. And the reason is the unawareness. We are not aware. We are not aware of what to do when we feel that there is something wrong with the health, right? Where to go? Insurance is there or not? A lot of things. So what happens when we talk about breast cancer? If you see the breast anatomy, just put it here. Yeah. When you see the breast anatomy, in a nutshell, or we can say that easy to understand, breast consists of duct, lobes, fat, and pectoral muscles, right? So duct produces milk through nipple, right? And then we all are blessed to have immune system, strong immune system, because we were fed milk by our mother, right? So that is the best immunity that a baby can have. And unfortunately, when you see the breast cancer, 70% of cancer occurs in duct. So ductal cancer. Lobes is called lobular cancer. If ducts and lobe is called mixed tumor cancer. If it's inflammation, it's called inflammatory cancer. If there's a mucus, it's called mucinous cancer. So this is how the cancer types are divided into several types. The way to understand, so these are some of the videos. In fact, I will just share this thing because I don't want to take a lot of time. These are from Nucleus Media, so you can always go back and see in YouTube about breast anatomy and how the breast cancer stages are defined. So now, other than that, what we need to understand is that there are two types of breast cancer. One is non-invasive, one is invasive. Non-invasive is when the breast cancer or the cells would be in situ in the same area. It will not move to other area. There's an invasive when the cell moves from one area to other area through lymph nodes, right? So it's called invasive cancer. If the invasive cancer is there in ducts, it's called invasive ductal carcinoma. If it occurs in lobes, it's called invasive lobular carcinoma. If the inflammation is called inflammatory breast cancer, and there's a pancreatitis. So highest, as I said, is 70 to 80% in ducts, which is this a particular area, as you can see here in this left side picture. So the point is, can we so now let us understand this much we understood, right? Now let us see the diagnosis flow. So first is that self-examination. All the women are advised to do a self-examination once a month to see whether if there is any inflammation or there is any kind of lump or there is any mucus, right? And if there is that kind of thing, then the person has to go to a doctor where there is a clinical breast examination. So the clinician will test whether everything is okay or not. If there is suspicion, then a person has to go for mammography or MRI. When person goes for mammography or MRI, if the region is suspicious, like you can see here, right, the person is asked to go for breast biopsy, where the tissue is taken out, and then the tissue is studied for different biomarkers to confirm whether the person has cancer or not. So this is the flow. Another important thing is every year for women between 40 to 54, they should go for mammography, where above 55 and above, they should go every 2 years for a mammography. Do we know this? We don't know that, right? A lot of people don't know this because of the lack of awareness in this particular domain. If we are aware and if we can catch the breast cancer at earlier stage, the chances of survival would be extremely high. That's why when we see the bar on the left side, in the previous slides, what we were looking at, the incidence is very high, but also the survival is high, the mortality is low. So that is something called awareness. Now, let us come to the point that if we have this tissue and if we slice the tissue, this tissue goes to the pathology where they look at HNE, estrogen, progesterone biomarkers. Now, the question is that this takes about 2 to 3 hours. So when the doctor is operating a patient, right, if there is suspicious and then the person has to go for operation, surgery, and when the doctor is operating a patient, at that time, when the tissue is taken out, they have to send it to pathology lab to understand that whether complete cancer is extracted or resected or not. It will take 2 to 3 hours for them till the patient has to be in the operating theater. So can we develop a technology that as soon as the tissue is out, we can place the tissue in this, using this technology, we can tell there is cancer or there is still, the complete tissue is taken out or there is still normal present in the tissue, right? Or there is cancer still present in the tissue. So to do that, what we suggested is, can you give a slice of tissue and can we let us develop a technology to work on the same. So that's what this is the workflow. If you see the assessment, like I said, clinical examination, ultrasound, X-ray mammography, and then surgery is done. When the surgery is done, this is the where I am talking about. When the surgery is done, they have to send it to histopathology. Wait till these reports are back and then they have to continue the surgery. So if we can develop, because you see this example, if this is a tissue, it's fine, the complete tissue is out, but if still there is a cancer, then they have to still go for surgery and remove this cancer at all completely. right? So what we need to understand is, can we develop a technology that can tell it is cancer or normal? This is the same graph in a different way, right? You can see that the 9.9 million deaths because of breast cancer. So what we propose is, can we understand the electrical property, thermal property, mechanical property of the breast tissue, and develop a technology that uses these three modalities, electrical, thermal, and mechanical, to come up with a diagnosis that whether the tissue is cancerous or not, and that can be used in an intraoperative environment. So that's what the flow cycle currently is this one, where my laser is fusion section, extraction, microscopy, and sectioning of image and telling the diagnosis. What we suggested is, when the tissue is extracted, give it to us. We have a system that can tell based on electrical, thermal, and mechanical properties whether the tissue is cancer or tissue is normal. So to do that, we divided the work in three different parts. The first part is electrical and thermal studies. The second part is biomodel electrical transport studies. And the third part is on electrical, thermal, and mechanical studies. So let us go with one by one. But in all the studies, the important thing is your sensor, right? So I'm not sure how many of you know the microfabrication, but irrespective of that, let us go to the final chip, right? So this is a process flow for fabricating this microchip. Let us go to this particular chip. If you see my laser, this is a left bottom figure, right? Schematic. In the center, there is a micro heater. All right, in the center, there is a micro heater. Surrounding the center region here, where my laser pointer is there is an interdigitated electrode. Then there is a trench, there is a pit here, and then there are three thermistors or RTDs. You can see here 1, 2, and 3. Now, why there is a pit? Because if I heat the heater, right? If the heater is at high temperature, that should not cause change in the RTDs, because I don't want to see the effect of this temperature changing the RTDs. I want to see the effect of tissue placed on this particular chip, right? And understand the thermal conductivity. I'll tell you about that. Let's not worry about that right now. This is actual fabricated chip, right? These are contact pads, and the sensor is right in center here. Or sensors, we can say two sensors at least. One is your heater with RTDs, and another one is a interdigital electrode. This is an SEM image of the same. Now, generally, what we do, this is an optical profilometer, as you can very clearly see to understand the thickness. Now, most of us will go for wire bonding or will go for soldering, right? So in the area of biomedical engineering, when we are going to use biosample, it is advised that the chip is discarded after every experiment, after every data collection. We need to take out the chip, throw it, put the new chip. To do that, we thought about why not to go with a technology similar to our SIM card. If you see SIM card, any person can easily insert it, right? So we made this slide field contact. Of course, there is a PCB that can take this one, and then there is electronics. I'll do that because there is change in resistance that we need to see, change in the temperature that we have to see. I'll talk about it in detail. So these are the engineering drawings. Now you do understand this thing because this is the heart of the entire technology. So of course the chip that is there, right? We insert this chip. One chip is here where my laser point is there. I hope that you can see my laser point on the right side. On the left side, there is another chip attached to it. So it is here and here like this, okay? In between, you can place the tissue. In between, you can place the tissue. These both are like a robotic arm, you can say, or manipulator. And when you press a button, there's a UI that we have developed. These both manipulators will come together. So let us understand this thing. I was talking about mechanical engineers to work with mechanical engineers, to work with computer scientists, to work with electronics engineers, right? So the reason you can see now, this is done by my student who has an engineering degree in mechanical engineering, and that's why the way it is shown is is how it should be shown: isometric view, top view, details about the CAD drawings, and then we have fabricated this thing. So let me just go through this actual photograph of the system. This is a schematic. Let me go to the next slide. I'll play this video because it's very important. After this video, I'll tell you what happens. Okay. All right. So you were able to see there is a chip here, there is a chip here, and then there is a indenter here, right? So we'll talk about this thing and in some time. Let us focus on this chip and this chip. So there is a tissue in between, right? So what was there on our chip? Our chip was having heater and RTDs. So if I heat the tissue in this side, right, what will happen? That there is a temperature difference here, right? Because you are heating chip from one side, the RTDs are changing on this side, so there is a temperature difference, right? So from T1 and T2, if you heat the tissue on one side, understand the change in temperature on the other side, what you can understand, you can measure the thermal conductivity of the tissue. Now, if you remember, our chip was having interdigitated electrodes. So if I apply a voltage between this electrode and this electrode, what will happen? There will be flow of current, right? Flow of current depends on the resistance of the tissue. Another thing, there are interdigital electrodes, means I can also apply voltage between the electrodes and measure the surface resistivity of the tissue. So what I can do, I can measure the bulk conductivity, thermoconductivity, bulk resistivity, and surface resistivity of the breast tissue. So here I can understand electrical property and thermal property. So this is how the tissue looks like. We have a collaborator from Assam Medical College. We have got the ethical clearance to use the breast tissue, formalin fixed tissues, and we also have the ethical clearance from IISc, and we also have Institute Biosafety Committee approval to use this tissue in the laboratory. So these are the gold standards, IDC and adjacent normal. Same tissues are shown here. And then you can see here the change in the bulk resistivity. What we were able to see, this is very interesting results. If you see, we have taken tissue and measured the bulk resistivity. All right, we start with normal tissues. How many subjects? 1, 2, 3, 4. 4 subjects. 3 times we had to do the study to get the biological lipids, and we can also get the standard deviation. And then we heated those normal tissues at 37 degrees centigrade and measure the bulk resistivity, right? How we can heat? Because you have the micro heater. So if you heat the tissue at 37 and then you are again measuring bulk resistivity, then we were able to see the change in the bulk resistivity of the tissue at 37 degrees centigrade. Same thing we did with 25 degrees centigrade for cancer tissues, and we heated the same tissues at 37 and measured the bulk resistivity. We were able to see a significant difference between the tumor tissues at 25 and 37, while for normal this was not there. It was non-significant. That means that just using a tissue, we don't want to do a lot of studies. If you take a tissue and you heat the tissue, you measure tissue at room temperature and you heat the tissue at 37 degrees centigrade, if there is no change or the change is not significant, you can say it's a normal tissue. When the change is significant, you can say it's a cancerous tissue. So this is the finding that we found. We also find a patent that is a separate thing. The paper is under review. But the interesting thing was this thing, the effect of temperature on the bulk resistivity. Same thing we worked with the formalin fixed tissues. So deparaffinized tissues is when tissue is taken, it is kept in a wax and it is stored in the tissue bank. When you want to reuse the tissue, you have to remove the wax. Removing the wax, so dipping the tissue in the wax is called paraffinized or paraffinization. And removing the wax from the tissue from the wax is called deparaffinization. Xylene is a material which is used for deparaffinization. All right. So I hope that this much is understood. Deparafinization and formalin fixation. Formalin fixation is when the tissue is taken out, it is kept in formalin to preserve its structure, right? Because normal tissue cannot stay for a long time in a room temperature. So we were able to see a difference. Now we were also interested in surface resistivity, as I told you, and here also we were able to see a difference in the normal tissues versus tumor at 25 and 37 degrees centigrade. And then further, when we looked at thermal conductivity, that means when we are heating the tissue from one side, measuring the temperature on the other side, depending on the temperature, we can measure thermal conductivity, which was, we were able to see that in case of the normal tissues, at 2537 or cancer tissues at 2537. When we talk about normal versus cancer, there was a significant difference between the thermal conductivity of normal tissues versus cancerous tissues. This was one part of the study. Now let us understand a little bit from the modeling point of view. So we were interested in understanding the biomodal electrical properties of the tissue. And for that, what we were interested was to understand what are the scaling laws and whether the scaling laws can be used for understanding the electrical transport in tissues. We also wanted to study the biomodal frequency and temperature dependent electrical transport studies of human breast tissues. So until now, as per our understanding, these laws were only used for composite materials. But since our tissue is a disordered structure, having disordered structure, we wanted to study the scaling laws. Now, if you all know, scaling laws are functional relation between two physical quantities that scale or increase with each other over a period of time, right? So when you see the normal tissues here, right, versus fiber adenoma, which is going towards cancer, what we are able to see is that the normal tissues have longer collagen fibers. These are, you see these green color fibers, these are longer, right? And lesser in number. Because of longer, the number of fibers are less. This leads to high disorder state. This leads to less disordered state, right? Because less number of fibers and longer in structure, less disordered. If you see the cancer or tumor, what you are able to see is these fibers, collagen fibers are higher in numbers and they are shorter in length. This leads to a higher disordered state. Now, because there is a disordered materials, that's why we can use the scaling laws. So what we did, we use the same thing. We use our system. We measure the impedance, and I'll tell you how we do the experiment. You can see here very clearly the chip is attached to a PCB which is connected to the indenter, and when we press the tissue, right, we were able to see the change in the conductivity, and we were able to see the effect of normalized conductivity with respect to frequency. So in this, what we can see is, you see this is red one is the tumor, blue one is the normal, and what we were able to see is that for frequency dependent AC transport, the AC conductivity scales as function of frequency, right? It scales as function of frequency. There is something we define as an onset frequency, all right? Onset frequency. You see, suddenly there is a change in conductivity. So the onset frequency is defined as the frequency that marks the onset of AC conduction. So shorter lengths of conducting filaments enables the electronics to transfer. So see, this is a shorter length, right? This red one is cancer, shorter length. So what happens that if there is a shorter length of filaments, the electrons transfer energy more effectively to the next filament, right? At higher frequencies. While since tumor has shorter length fibers, the tumor samples have earlier onset frequency that you can see, resulting in lower onset values. So the very simple thing is that we can see that the tumor samples would have a higher or earlier onset frequency. Another thing that we measured is conductivity with respect to temperature, 24, 25, 26, 27, 28, all the way to 37 degrees centigrade. And what we defined as a critical temperature from this study, you can see critical temperature is at which the collagen fiber starts degrading, resulting in lowered volume fraction. Since tumor tissues, which is the red color one, this one, right, already have higher extent of degradation, and disorder, they have lower critical temperature than normal. So you can see that they have lower critical temperature compared to the normal tissues. It's not already going in this particular manner. In any case, what we further understand and what you can see here is 15 kilohertz and 3 megahertz. Why we selected these two frequencies and not other frequency is that the 15 kilohertz and 3 megahertz cover the entire AC conduction region for the majority of tumor and normal samples, and that's why we used this particular frequencies to understand the model. We have fitted the model, and you can see it's very well fitting, right? We have tried adjacent normal versus tumor. We measure the R-square values, and we measure the change in these the critical temperature as well as onset frequency. This article is also under review. In fact, it is a minor revision. It should be accepted anytime soon. So let me go to the another step, and then I will stop for some time to get the queries from the audience, and then we'll continue to the next topic. So as I told you that we were interested in understanding electrical, mechanical, and thermal. Right now, what we have studied, we have only studied electrical and thermal. So let us see that what happens when we add one more modality, which is mechanical. And mechanical means understanding the stiffness of tissue. So this is a representative diagram of one of our paper, which says that the electrical impedance for normal is less than tumor, the thermoconductivity for normal is greater than tumor, and the stiffness for normal is less than tumor. So in this case, we also used a piezoresistive sensor. This is a chip that you can see here, right? This already you have seen earlier. This is a new chip that you are able to see now. And here we have 4 piezoresistive bridge. You can see is a single area is a piezoresistive bridge. How to design this or how to fabricate this? This is used using the diffusion technique. So if you take a N-type silicon or polysilicon in which you can diffuse boron, right? Then you can fabricate piezoresistive bridge. Piezoresistor, as we know, when we apply a pressure, there is a change in resistance. Is different than piezoelectric, because when in piezoelectric, when you apply a pressure, there is a change in voltage, right? And there are 3 things here. There's an electrical sensor, and then there is a chip that is attached to which there is an indenter which is attached. Okay, there's a chip here, which is this particular chip, right? It is right over here on my, if you follow my laser pointer on the right bottom, or you can see this schematic also. This is a chip here. This is a force sensor. This is my electrical and thermal sensor. This is a tissue, and then there is an indenter. All right. So now what will happen is, when I know electrical and thermal, right? But if I apply a force here, then I can measure the stiffness of the tissue. So we were able to understand the change in the electrical properties. We were able to see the when you indent the tissues till 1000 micrometer, which is 1 millimeter, right? We were able to see that the normal tissues indentation load is different compared to the tumorous tissues. And from that, we were able to see that if you add 3 different modalities. You see, just electrical modality will give a separation, will give a delineation between normal and tumor. When you add a thermal modality, it is also showing stiffness is also showing the change, right? You see here in this particular plot, right side. But when you merge all 3 modalities, then the significance is higher. The p-value is decreasing, 7.24 e to the power minus 8. That means that adding 3 different modalities into a study will help us to understand a system that can delineate normal from tumor once the tissue is given in the intraoperative environment. We have done these studies for about 32 subjects. We have to do this study for close to 500 to 600 subjects to make sure that the system works very well. There is an effect of BMI. We are looking at that. We have to look at the fresh tissues. We are looking at the collaboration. So a lot of things are there to work further. This is a system that we can use after the tissue is taken out. So we are also developing a tool that you can use in vivo. What is in vivo? There are 3 things. Okay, so let me stop the screen right now, and I didn't share the screen. There are 3 things. One is called in vivo. In vivo means within the body. When you take out the tissue and you measure, it's called ex vivo, which we have seen right now. Third thing is in vitro. When you take out the cells, develop the tissue in the laboratory and studies those tissues, it's called in vitro. right? So right now, whatever I have shown it to you in few slides was the study of ex vivo breast tissue or breast biopsy tissue, because tissue was small, to understand or to delineate normal from tumor based on three different modalities: electrical, thermal, and mechanical, right? So I will now move ahead and talk about the design and development of a micro force sensor with haptic feedback for catheter that can be used in atrial fibrillation. But before I go to that topic, I would like to see if you have any question. If not, we'll continue.
So participants, any questions? You may ask Dr. Pandya.
Yeah, I think they are all locked, no? So they may not be able to ask. So if they can chat, right? In the chat box, they can place the question.
They can be able to, they can chat, talk, sir.
Yeah. Oh, I see. Okay.
Sir, I have a question. Um,
Yeah, please.
Cancer have different stages in stage 1, stage 2, stage 3, stage 4 like that.
Yes.
Does the characteristics means is electrical, mechanical, or thermal characteristics changes with the stages, or it remains constant? There is any change
Very good
in
question, Siddhartha. Yeah. See, the idea is stage-wise study is already done in the pathology lab. All right. So there is no gap in that. That means that they can understand if stage one is there or two or three or four. The gap is right now in the intraoperative environment. That means the patient, the clinician has to wait till the path lab comes back with the tumor. So our technology is to understand. There are two ways. One is to understand when the tissue is out, whether it is normal or tumor, without considering stage is there or not, because this tissue is taken out from the surgery. Stage and other thing comes before the surgery is done. All right. So that is one thing. Second thing is the tool that we are developing is in vivo. That means during the surgery, a surgeon can use the tool to understand there is a difference between normal or tumor. So the whole focus was on delineating normal versus tumor, not on understanding the stages. However, if you are interested to look at the properties, we have done studies, not in my lab, but previously I have done to understand the difference between different stages like benign, no, ductal carcinoma in situ, lobular carcinoma in situ, invasive ductal carcinoma, and invasive lobular carcinoma. So rather than stages, types of cancer is what we have studied. You can study out of curiosity to understand if there is a difference in the stages. But what I understand is from the path lab, which is the gold standard, stages can very easily be identified.
Okay, sir. Thank you, sir.
But yes, it would be interesting to study if one has to study the different difference in this ETM properties stage wise.
Yeah. So any other further questions from the participants?
It will not be so easy also to understand some of the
Yeah.
things, right? So let them take time. If you have questions, do contact the host and co-host, right? And then I'll get back to them with the list of responses to the queries that you may have at later point of time. So should I continue my lecture, Alash?
Please, sir. Please, sir.
Yeah, sure. Okay, so I'll stop video once again and I'll continue with the next set of work, which is on micro-engineered force sensor and haptic feedback system for catheter contact force measurement. Okay, so let us understand what is catheter contact force measurement. Before we understand that, let us see this schematic. In the left side schematic, right, you can see a patient lying under the X-ray. There can be MRI also, X-ray also. There are MRI compatible robots nowadays. So anyways, the point is that generally human heart beats around 65 to 70 beats per minute. When arrhythmia is there, then the heart starts pumping faster. It starts pumping faster. So if it starts pumping faster, number of beats would increase. There is one disease which is called atrial fibrillation, where the heart starts pumping unevenly. This uneven pumping is because of the misfiring of electrical signals. So to understand or to treat that particular disease, which is atrial fibrillation or AFib in small, right? AFib. The way to do is that the surgeon or the electrophysiologist would insert a tube called catheter into the heart. Mostly it is done via the groin area, because that's easy to insert the tube, and this tube is called catheter. Okay, this tube is called catheter. It reaches to the area of the heart, and then first one is electrical mapping. So there are two catheters. One is only to understand the electrical signals originating from different part of the heart. So it goes inside and it maps the entire heart to see that where exactly the misfiring of signals are happening. Once electrical mapping is done, this catheter is drawn back and another catheter is placed inside, and the area which was misfiring, right, that area would be burned. The burning of that area is called ablation. Now, to burn this area, there are two ways. One is called cryoablation, another one is called RF ablation. So if you see, there are two companies right now. There are few companies that are working on the same, but Techicat and another company which works on the developing this catheter tubes, right, with the electrodes to ablate the heart tissue. And there is holes here. You can see that, right? There are holes here. These holes are for saline irrigation pots. What is irrigation pots or irrigation holes? Is that when the catheter is inserted, there is a flow of blood, right? There's a flow of blood. So the catheter, through the catheter, we can insert saline. So it will make a space. It will make a space, right? And saline can be flowed through this catheter, through these holes. Now, when you want to apply a radio frequency, you need to touch the tissue, isn't it? So to touch this tissue, the catheter is placed in contact with the heart tissue, and if the force is more, other regions of the heart will burn, isn't it? If the force is less, then the recurrence will occur. Recurrence means patient will have the similar episode after the surgery is done. So the very important point is to understand the optimized contact force. That optimized contact force, already we know from the literature, it is 0.35 Newton, which is about 35 grams, which is optimized force. Now, this force can be seen on the screen when the surgeon is operating, but the surgeon would not be able to feel the force. The feeling of force is called haptics. So there are no commercially available catheter that gets the haptics feedback, and if you can add these haptics, the surgery will become better. It will facilitate the surgeon to operate the person suffering from AFib. So with that goal in mind, we started working on developing or fabricating a catheter with a tip integrated with a force sensor and the related haptics. So this is a force sensor that we designed. It has 4 piezoresistor on one bridge. There are 4 bridges, and then there is a structure, like you can see here, some indenting structure that indents only these bridges. All right, not really bridges, but the circular part that is holding the bridge. So if I apply uniform normal force on this, all this bridge will uniformly bend. When it bends, there is a change in resistance. This corresponds to the force that we have applied. If there is a normal force versus shear force, we can see a corresponding change in the resistance of each bridge. These sensors are microforce sensors created or fabricated using microfabrication technology. This is an actual SEM image of the force sensor. that we have designed. This is how force sensor looks like in compared to one cent. And you can see here there are two types of force sensor, one without any holes in the bridge, second one with the holes in the bridge. The reason of creating this hole in the bridge is to improve sensitivity. So we start with designing the sensors, we go for simulation, modeling, then we do fabrication, then we develop this catheter tip, then we integrate it to a catheter, then we use for issues, and the experimental data, along with all this data, we publish as a part of a journal paper. So this is how we are working on in the lab. Now, to first understand or characterize these force sensor, we have wire bonded this force sensor on a PCB, and then we have indented by applying a normal force to the catheter. As you can see here, directly this indentation is on single bridge to see how sensitive the sensors are and how the bridges are changing. So when you indent each bridge separately, you can see the change in the resistance with respect to the force that you are applying. How you know how much force you are applying? Because this indenter is connected to a commercial available load cell. All right. So once you have that, we design a tip. Is a tip on which the sensor is integrated. This is a PCB. This is a sensor, and then there is a wire bonded, and this whole thing is integrated onto this particular PCB. And then to this PCB, we have indented a load. You can see here, tip, right, which is indenting or applying a force on tissues. These are porcine tissues or pig's tissue, pig's heart tissue. Anatomically, pig's heart is close to human heart, and that's why we have used pig's heart. Again, we have got the Institute Animal Ethical Clearance to use porcine heart. We also have Institute Biosafety Clearance to use the heart in the laboratory. We also have Institute Human Ethic Clearance to use human heart. We are on that particular stage right now. But this is what I'm showing is the force sensor, the role of force sensor with the catheter tip to understand the change in the resistance of each bridge and correspondingly to develop a catheter with a haptic feedback. So here we have done several experiments I was telling you, right, that we have a structure that can directly indent this circular structure of the bridge or of the chip. Here also you can see, and there is a normal indentation versus there is an angular force. When we do this angular force versus normal, there is a change in the resistance correspondingly, which we can very clearly see from the data obtained using our system. This is where we started developing the sensors. There are about 16 the contact pads, because 4 sensors on one bridge, we have 4 bridge, that results into 16 sensors. These 16 sensors we have to integrate along with all these things into a catheter tube. So we started with developing this particular structure, the indenter mechanism. Again, you can see engineering drawings, right, to indent it. We have done study with the to develop electronics. For the same, we used ERM motors, because finally the idea is not just to measure the contact force, but to also understand the haptics along with that. So we have this ERM motors right here. You can see the entire closure. There's a vibration motor, silicon cap, fasteners, spring, rubber garments, and then SS screw to fit all this thing into each one. So you have, if you put 4 fingers, right, you can understand the change in the vibration of this motor corresponding to the force that is applied on the heart tissue. From that particular stage, now we have this catheter that you can see with this ERM motors integrated onto this handle, and this can be connected to a system where we can see the force, but we can also feel the force, right? I told you that with the commercial catheters, we can see the force, but we cannot feel the force. With this catheter, we were not only able to see the force, but also to feel the force. So this is where we are in terms of designing and fabricating a catheter with a contact force measurement. Contact force is this tip contact force on the tissue. So we have done some study with the entire catheter, and we were able to understand the difference between LV and RV for different force range. This is LV, RV is a region in the heart, the tissue from the LV and RV. And then as you see, the intensity would increase depending on the force that you are applying. So those are the results in this particular slide, that this intensity will be corresponding to the vibration of the motor that we have integrated onto the handle of the catheter. Now, before stopping here, I'll go with one more subject, and then I'll take question, and then I'll continue the discussion. Okay. So this is on another character. First, let us understand the current technology. This is for understanding the tracheal inflammation in infants. Now, infants, when they go to hospital and if they are on ventilator, the trachea is a windpipe. That windpipes gets inflamed. So when windpipes gets implemented, there is a way to check it, and the way are shown in this particular slide. First one is endotracheal tubes. This tube is sent, and then there is a balloon. You apply air, and this balloon will expand. If it is not fitting, you will take out the tube, put another tube. So there is lot of interchanging of tubes that happens. Second is called Kirschner wire, which you can, like this measuring stick, like Allen key. You just put it directly, and whichever fits, you can tell this is an inflammation. Right. Third one is bronchoscope. It depends on how good your doctor is, how well it can see, but optical distortions are creating a problem. That means that you need to interchange a tube, or the sticks needs to be swapped until right fit is determined, or the optical distortion has to be taken out, and severity of narrowing are often misclassified. So there is a gap in the current technology, and those gaps we can overcome. Now, another way of checking is by asking a patient to go for spirometry. Another way is a bronchoscopy, as we have discussed, and then tracheostomy, and then there is a radiological scan. So, lot of things, but you understand that infants, when you ask infant to lie down in an MRI, it's not possible, right? So, lot of difficulty with the current technology, as you can see here. Diagnostic methods on left side, challenges on right side. On diagnosis methods are bronchoscopy, echocardiogram, MRI, CT, tracheobronchography, pulmonary mechanics, right? And the challenges are bronchoscopy requires general anesthesia, the patient cooperation in providing inspiration expiration. For a child, it's very difficult to ask child to inspire expirate as what we want, right? So it's very difficult. Sometimes a child is incorrectly diagnosed with severe asthma, where they really have the tracheomalacia. So this is very important thing that we need to understand, that the wrong diagnosis is very difficult to accept, because if you don't have asthma, and the process for treating starts for asthma, but actually it is tracheomalacia, which is the flaccidity of the supporting tracheal cartilage or softening of the tracheal cartilage, right? The diagnosis, the treatment is different when it is tracheomalacia, which is the softening of the cartilage, versus asthma. So that is the reason why it is important that your child's physician has a lot of experience diagnosing and treating this condition. Several way and techniques. I'll not go into too much detail, but let us understand some of the terminology, right, that we will be using it. So there are a lot of challenges. The point of showing this slide is that in the current technology, the current diagnosis, there are several challenges. So yeah. So what are some of the techniques? So the one is called stenosis. What is stenosis? Stenosis is the tracheal narrowing of the windpipe. When there is a windpipe narrowing, it's called stenosis. Tracheomalacia. We were talking about tracheomalacia just before this slide, right? And it's a softening of the supporting tracheal cartilage. If you don't want to use softening, and if you want to use a little bit interesting word, you can say flaccidity. All right. Tracheostomy. What is tracheostomy? Surgical procedure where an opening is created in the windpipe for tube insertion. I'll show it to you. This one. This is a tracheostomy, right? So because there is a difficulty, so there is a opening is created where the tube is inserted directly into the windpipe like this. All right. Then there is a TIF, which is called tracheoinnominate artery fistula. This fistula is an abnormal condition between the artery and the trachea. If there is a integration of trachea with artery, there is a problem. Last one is called esophageal fistula. Esophageal fistula, TEF, is an abnormal connection between esophagus, which is a food pipe, and the trachea. So windpipe and food pipe should not be connected. You know, right? Sometimes if the food goes into the windpipe, we start coughing, and then we said, oh, it may went into windpipe. So food pipe and windpipe should be separate. So if there is a integration or there is a abnormal connection, it is called TEF or tracheoesophageal fistula. All right. Now these are some of the terms. Let's not worry about it. The point of our interest would be that how we can design a technology or a tube or a catheter that can be used easily to understand the inspiration, expiration, and in general the inflammation of the trachea, which is we call stenosis. So we designed a catheter, which is a tube with a flow sensors. You can see here 1, 2, 3, and 4, and with a SMA, which is a smart memory alloy, and with a force sensor that will be integrated onto the tip. If you see the trachea, and trachea generally is a D shape. That this is a bone, and this is a you can see my laser pointer, right? So this is one side of the tissue, and this is another side of the tissue. So we need to know if there is a stenosis like this. See, it's inflamed. So if I place the trachea inside and a person inhales and exhales, then that particular flow sensor will show change. Now, what is this flow sensor? It is just a thermal flow sensor. That means it's a micro heater. So if I have a micro heater, I can see the change in the temperature depending on the airflow. The change in the temperature in this particular heater would be different compared to the other heaters because there is a stenosis in this particular region. Now to manipulate or not manipulate, not to maneuver the catheter, we require a smart memory alloy. If you know, it's called SMA or smart actuators. So we have integrated that into the catheter. We have this micro heater fabricated using microfabrication technique. One mass process, very simple. Platinum is used along with titanium as a bottom. And you can see on a 4-inch wafer, you can get hundreds of micro heaters. This is a flexible PCB that we have designed. And the idea is that this PCB is made up of PLA, right? And also, PI, PI and copper tracks are there, and it's a double layer PCB on which we can integrate this particular micro heaters, and we can do wire bonding. Also, at the tip of this PCB, we can insert the SMA, and we can have the force sensor. I'll show it to you if I have the slide for the same. Now, what we have done is first understood the microflow sensor. We inserted a tube in a sheep trachea. Sheep trachea we started is an animal experiment. So from the dead sheep, we took the trachea, we brought to the lab, we have ethical clearance, animal ethical clearance for using this particular subject. And we also have institute bioethical clearance to use the tissue in my laboratory. I'll show you some of the facilities that I have in the department, and remaining facilities you may know, which is central facility of the institute, which we have a fab lab. A small fab lab is also with me here in the Department of Electronic Systems Engineering. So the point is, when you don't have any stenosis versus when you have single stenosis versus you have two stenosis, then the pattern would be definitely different. 3 stenosis, right? In this case, you can see that the changes are different. In all the cases, you can see the signature from the microflow sensor or heaters is different for different flow rate of the air. And here you can see the inspiration expiration cycle. This we have done using the ventilator that is we have created here in my lab, and we were very easily able to see the change in the voltage versus force. We have also designed this force sensor. You can see here, right? Each force sensor has multiple piezoresistor. This is the backside of the force sensor, which is to create the pit using DRI. And you can see here that there is a different cartilage, radial cartilage, cranial cartilage, median cartilage has a different values of force when you indent it compared to the smooth muscle. From this, there is a literature which shows that from this particular data, we can measure what is the stiffness of the tissue. This paper has already been submitted under review, and the work has been patented as well. Now, before we go to this particular topic, right, I wanted to get like 5 minutes break, but before we go for 5 minutes break, I'll also want to see if you have any questions that I can answer, and then we will go to our next topic, which is on brain computer interfacing. Several implants I will show you, the micro electrodes or micro needles, flexible devices that we can implant in rat's brain, and then a tool to understand or delineate normal from tumor in brain cancer. This all things integrates sensors, as you can see, it integrates electronics, as you can see, it integrates mechanical engineering, it integrates your 3D printing, additive manufacturing, right? And to an extent, a bit of machine learning. If you have seen, I've not covered a lot of machine learning in this particular lecture, but let us see the next set of slides after I take the questions, if you have any from one of you. Otherwise, we will continue. Yeah.
Yeah, participants, any questions?
Hello.
No questions from the participant sides. Okay, sir. One question, just a small question, because this topic is completely out of.
Yeah, someone was asking a question.
Yeah, please.
Sir, how you got the idea of using?
Yeah, Akash.
Yes, sir. Sir, I am from thermal engineering in NIT Warangal. So my question is, what is bronchoscopic form? Sir, what is bronchoscope? How it is applied? Yes, sir. Sir, bronchoscope.
Yeah. So your question is what is a bronchoscope? Is it? Yeah, it's like a tube which can be used to treat lung problems in like tumors or bronchial cancer, right? If you see, you can see a lot of details about what is bronchoscopy and how it is examined. Generally, what happens is the airway passages are blocked, and it's like a tube-like instrument with a light and lens for viewing that area.
Yes, sir.
So a doctor can very easily see how when they will insert the tube, they will be able to see how the region looks like, and from that they have to understand that how much is the tissue inflammation. All right. So that is all the bronchoscope is. It is used to understand the inflammation and also for several lung diseases.
Okay, sir. Thank you, sir.
Sir,
Okay.
sir, how you got the idea of using electric like conductivity and resistivity to identify breast cancer, sir? Like, was there any incident happened, or how you got that idea, sir?
No, not for getting an idea. Not every time incident should happen, right? Generally, we have this thing that, oh, someone I saw and then this happened. No, it's not like that. I started working in my postdoc in Maryland, and the project was on breast cancer. When we got the tissues, we went for SEM studies. SEM is scanning electron microscopy, and there I found that the morphology of the normal tissues are different than cancerous tissues. So our initial interest was only to develop a cantilever to understand the stiffness of the tissue. But since the morphology was different, morphology means the structure of the tissue was different. If the structure is different, being from electronics background, I thought that if the structure is different, there would be some difference in the resistance of the tissue. And if there is a change in the resistance of the tissue, then there should be some difference in the thermal conductivity of the tissue. So why not to develop sensors to measure not only the mechanical properties, which is a cantilever to measure stiffness, but also to understand the impedance or resistivity and to understand the thermoconductivity. That's how the seed started. And then whatever I have done in my earlier work, my PhD student has worked 10 times better than what I was able to do at that time, right? So we are at very advanced stage in this particular technology. The things that we are waiting is large set of samples and fresh tissues. So that is what I got an idea from Prashant. Prashant was there, right? Who asked question?
Yes, sir. Thank you, sir.
Yeah, no problem, Prashant. Yeah, I think Palash, you were asking something, right?
Yeah, it's a very basic question, sir. Like on slide number 38, you have shown some simulation results and experimental results, sir. So, like on what softwares you simulate all these the experimental thing?
Yeah, COMSOL multiphysics.
On console only.
Yeah.
Fine,
so Yeah, the theory we of course do math, then we use COMSOL to simulate it before we go for fabrication. After simulation studies looks interesting, we go for because simulation study will tell us how close we are in respect of we fabricating a device with certain dimensions. Once we are sure, we go for fabrication, and once we fabricate it, we integrate it with the system. We develop the electronics around it. If there is a requirement of printing, we do a mechanical engineering, 3D printing, and then we apply it for the actual studies. So there is a, in a way, it's a complete cycle, right from theory to experiments.
Okay, sir. Thank you. So any further questions from the participants? You may ask Dr. Pandya.
Okay. Yeah. Good morning.
Hi, Tagesh. I cannot hear you, Tagesh. Now it's better. Tagesh, still I cannot hear you.
Yeah, I hope now it's okay.
Now it's better. Thanks.
Yeah, so I was saying a very good morning. It was this is what we call as a cutting edge research. Actually, I have been following you on LinkedIn for quite a while actually, and it was a really pleasure to have you as one of our speakers. Okay, so my question is because I'm kind of a biologist, I'm trying to grasp the new techniques you are trying to put in. That the first thing is that you were talking about checking the cancer cell morphology versus a normal cell. So as a biologist, we understand there might be like changes in the proteins on the surface of the cancer cells. And of course, we really don't know. There will be like a lot of proteins which might be changing on the cell of the cancers. What I'm trying to say is that can we do a kind of a non-invasive kind of a sensor? Because right now, as you present, it's mostly it's a kind of an invasive one. We have to take out the tissue and do it. So is there any methods currently going on? Because there are a lot of proteins which are kind of like a signature proteins for the cancer cells. So can we take out this protein and put it on a chip and do it kind of a non-invasive method? The same way we do it for the diabetics testing.
Yeah. So just to little bit reiterate or clarify, the work that I have shown is on tissues and not on cells. So we don't go for genomic study, we only go for phenotyping. But answering your question, what you are asking is a minimally invasive, because still it is invasive. You have to take a biomarker and you have to study it whether there is a presence, right? So it's a minimally invasive technique like glucometer, what you gave an example for. So for that, if we want to understand protein, we have been working on a project from BDTD, it's a DST project, on understanding the presence of DSG3, which is a protein present in oral cancer patient. So what they do is generally when there is a oral cancer, they kind of remove the entire lymph nodes. Now lymph nodes are responsible for our immune system. So when they remove the lymph node, the immune system of a person goes down. Instead of that, what we proposed is that if you take a fluid from lymph node and you place the fluid on the chip that we have developed, can we measure the presence of DSG3 protein? So by looking at the impedance change. So we have the sensors in the lab that we are working on with the surface chemistry. We have Pexilin surface chemistry. We have different MUAK surface chemistry where we load the antibodies and then we put this different concentration of protein onto this chip to see how the impedance would change depending on the concentration. That's one. And if you have a specific antibody, then we need to understand if there is specificity is also intact. So you can, what you are asking, we can do it. In fact, one of my postdoc, she is working on understanding different protein in breast cancer. One of them is HER2. So we are using anti-HER2 antibodies and trying to capture those HER2 proteins. Right now we are doing with a PBS. Going forward, we will look at the blood and would like to see whether we can still capture those protein or not. So the answer to your question is yes, we can do it with a minimally invasive technique.
Okay. Hardik, so the second thing is that from where, do you have to take a blood or you have to take tissue samples for testing? That is the one question which has been running on my head for quite a while, actually, because I was also thinking about something like this, actually.
Right.
So what is your opinion? What's your take on this? Like from where we can procure the samples for such kind of a testing?
Yeah, so you should have a clinical collaborator. Fortunately, I have 22 clinical collaborators from different regions, right? So they have to take the ethical clearance from their hospital. Before you ask them to take an ethical clearance, you should have shown some kind of preliminary data that what we are doing right now, right? That, okay, we are using PBS, we are using the known protein, and then we are looking at impedance measurement. So from this study, we are able to see that the biosensor is working well. Now, this study can be presented in front of the ethics committee to see that because we have reached to this stage, can you give us ethical approval to use patient samples? And then you have to get the number of patients, which is based on the statistical analysis. There's something called power calculation, like how many subjects you want, right? So, but it is very much possible to go ahead with this kind of ethical clearance. Most of the time, you will get it through because it's not complex. You're just asking for a few milliliter of blood, maybe 2 milliliter or so, right? So the right approach would be to work with a surgeon who works in the area of breast cancer.
Okay. Yeah, thanks for the clarification. One final question I have, you were talking about the catheter and you are using the force sensors at the tip, right? Possibly there's a kind of a blockage is there, then the amount of force resistance will be more at the tip. That's how it works, right? Like I'm trying to figure it out.
No, so maybe I was not clear. So let us say this is a heart, right?
Yeah.
And my palm is a heart. And then first they insert the catheter and understand different electrical signals in this region. Now they will know that, okay, in the center of my palm, which is the heart, the electrical signals are very high, uneven. So they will take out this catheter, put another catheter, touch this area and burn this area. So if you burn the tissue, it's called ablation. So when you ablate the tissue, the electrical current will not flow because it's burned, right? So that is called RF ablation. That catheter is called RF ablation catheter. And to burn this, you have to touch the tissue. So that's why contact force is important at the tip. When they can apply the force, they can see the force on the monitor. But when you are using the catheter, they cannot feel the force. So adding sensor with a tube and feeling the force is something that we are working on.
Oh, okay. I was thinking like we are using this catheter to place a stent. Normally they use a catheter.
No, not stent. No. That is a different technique. Yeah.
Different technique. Okay. Now it's clear. Yeah. Thank you, Hardik. Yeah.
No problem. Yeah. So then, if you allow me, I can continue with one more or two more topics. Palash, is it okay?
Yeah, it's fine, sir. It's fine, sir.
Yeah, sure. Okay. So let us see something very interesting and latest research. Now, whatever I am showing, with a significant amount of confidence, I can say that whatever we are doing right now in IISc, in most of the laboratories around the country is as good as someone doing in Western countries, right? We don't anymore lag behind in understanding or developing or working on the research domain, right? We are not anymore relying on somebody to give us the ideas. Now, having said that, does not mean that we should not collaborate. We should definitely collaborate, but why not to develop system that can be fabricated here? We have everything within our country, right? So the only thing is to have a right set of collaboration and to understand where the technologies or where the systems are available. So having said that, let us see some of the micro-engineered devices and brain-computer interfaces. I'll go one by one. If you see the human brain tumors, right, there are several kinds of tumors, gliomas, glioblastoma, meningioma, schwannoma, and other regions are normal. Then there are different regions. So suppose you say this region, right, this on the exactly on the front is called frontal region. When you see, when you see this through eyes, The region which is working is this region on the back side of the head is called occipital region. Then this one in the center is called central region. Just above the ear is called temporal region. So frontal, central, occipital, temporal, right? Different region has their own work. You can see here language comprehension, sight, which is occipital, hearing, motor function, right? Muscle sensation, eye movement, mental functions, which is right in the center. So this is about the anatomy of the brain in a very simplistic way. Now, what is the problem right now, and why we are interested in this particular topic? The problem is that when there is a tumor, as you can see here, right, is a glioma, the surgeon will operate this patient, suppose is a patient, he or she will remove the part of the skull, which is called craniotomy, and then they will remove the tumor from this region. Now, to remove this tumor, which is of this size, they will remove this much section, like a bigger section of chunk of brain they will remove to make sure that a complete tumor is out. Now, as I told you, the remaining area of the brain is for several different functions. So they are losing the normal tissues while the surgery is done. And that's why it is very important to develop a probe that can tell or that can give us a very clear tumor delineation or boundary between normal and tumor. If that is the boundary that we know, then the surgeon can cut the tumor just outside the boundary and not cut the normal tissues. That is our idea of developing this particular probe. And as you can see here, is a smart probe integrated with several sensors. We are trying to develop PMUTs. PMUTs are called piezoelectric micro machine ultrasound transducers. We are trying to develop an array of those, so to image as well, right? Not just to measure the electrical, mechanical, acoustic properties, but also to image it. Acoustic properties we can measure when we develop ultrasound transducers. This is an actual photo of the human brain during the surgery. As you can see, this is the tumor that they need to cut. Very difficult to first of all see. Second is that in MRI, when you open the skull, there is a layer which you need to remove, right? And when you open that layer is called dura. When you open the dura, there is a CSF. CSF is cerebrospinal fluid. When this fluid comes out, there is a shifting of brain. So in the MRI, if you have seen that this is a region, when you open it, the region will shift slightly. If the region shifts slightly, the patient has to, the surgeon has to be very careful, right? So right now the way to do this resection or removal of a tumor is understanding the grading by chemotherapy and radiation. Go for MRI, fMRI, PET, ultrasound, direct electrical stimulation, electrocardiography, Raman spectroscopy, fluorescence imaging, and then finally chemotherapy and radiation. So what are the commercially available technologies that we need to see? And for that, we have compared some of the technologies available, right, with what we are developing here. So, one second. Okay. So if you see here, comparison of the proposed intraoperative probe to existing tool, intraoperative MRI, you can see this intraoperative MRI, you require this kind of function. Low-grade tumor can be delineated, it's rapid, but external agents are not required. Portability is very low, cost is very high, ease of use is medium. Raman spectroscopy is best because it can give a very clear delineation in a low-grade tumor. It's very important. However, it takes very long time because from point to point you have to move the probe, which will take hours to delineate the tumor boundary. External agents are not required. Portability is low. Cost is high. Ease of use is high because it requires expert to operate this system. Next is the fluorescence imaging, and in that low-grade tumor, we cannot delineate. It is rapid, yes. External agents like imaging means it is a secondary biomarkers are required, or it is called labeled technique. Label-free secondary antibodies are not required to create a fluorescence. Next one is portability is very high, so it's good, but the cost is low, which is also good. Ease of use is high, which is also good. But because it cannot delineate low-grade tumor, so it is difficult. Also, if it uses external agents, it is not advisable. So what we are developing a probe is a probe that can delineate low-grade tumor. It can be rapid and real-time. It does not require any external agents. Portability should be extremely high. Cost should be affordable. While ease of use, I have not said anything, is because until surgeon uses it, we don't know. We have a clinical collaborator from NIMHANS. We have two surgeons, in fact, many surgeons, but two I'll give name today, which is Dr. Vikas, and we have another surgeon. And we also have Dr. Sabri from Ramaya Hospital. We have Manish Beniwal from NIMHANS. We have Professor Anita Mahadevan, who is heading the pathology in NIMHANS. So a right set of collaborators with whom we are working. This is a design of the PMUTs with microelectrode arrays. We call PMUT MEA. And this is a system, that UI that we are developing, which can help this probe to delineate the tumor boundary from normal. Engineering drawings, we have filed a patent. What this probe can do, it can measure the electrical properties, it can measure the mechanical properties like stiffness, elasticity, viscoelasticity. It can measure the acoustic properties like attenuation, speed of sound, and like I said, if we are able to fabricate array of PMUT successfully, we can use it in imaging as well. Now, this is a system that we have developed to understand the stiffness and the electrical properties. This is for electrical properties of tissues. This is an actual human brain samples. We have got ethical clearance from NIMHANS. We also have applied for Institute Biosafety approval. Right now, these tissues, my student went to NIMHANS with the system. They measured all these tissue properties. And what we were able to understand is that the resistivity of the normal tissues and cancerous tissues are very different. And that's why resistivity can be used as one of the biomarker for delineating normal from tumor. Whether we go for formalin-fixed tissues, fresh tissues, it does not matter. These are the pathological studies where you can see the hippocampus, you can see putamen, thalamus, this white matter, gray matter, all these things. So the idea is that if we want to understand normal versus tumor for fresh tissues, we were able to understand different brain regions, we were able to understand difference between white matter and gray matter. We were very clearly able to measure it. We also fabricated a force sensor, as you can see, again with a piezo resistor, and use it to understand the stiffness of the tissue. This is a system that we have developed. The tissue is placed here. There is already a force sensor. This force sensor is right over here, and we have applied a known load on this sensor. Now this is a M. If you see this one like this, it's a M, right? You again follow my laser. It starts from here, goes here, indents on the force sensor, comes here, and comes here. So if I place the tissue here and if I indent it, depending on the stiffness, this will come up. If it comes up, the known load will be reduced, and that would be effect of the tissue on the force sensing resistance. So this is how we measured, and we were able to measure the difference between glioma, meningioma, squamoma, brain, motor cortex, gray matter, white matter, sensory cortex, gray matter, white matter, frontal gray matter, frontal white matter, and glioma. What we were able to understand is glioma has significantly lower stiffness compared to normal tissues. Right? It is a very different study compared to the breast tissues. In breast tissues, we were able to see that the cancerous region is stiffer than the normal region. In this case, we were able to see glioma, particularly one type of brain cancer, has a lower stiffness. Tissues of brain demonstrate viscoelastic behavior, and white matter regions are stiffer compared to the gray matter regions. So from that, there are several things and challenges. So which we'll be talking in some time. But let me go to another part of this brain computer interfacing chips, starting with the epilepsy. So epilepsy is something called FIT. We all know, right? And the way to treat this epilepsy is by using anti-epileptic drugs. Now, if you see about the 70 lakh cases of patients are there for epilepsy, and in terms of millions, it is 7 million, 58% increase in epilepsy surgeries. Now, why to go for surgery is that when the anti-epileptic drugs are not working, we have to go for surgery. And in surgery, the part of the brain which is misfiring electrical signals, we need to remove that part of the brain. When I say we, is of course a neurosurgeon. So that particular type of epilepsy is called intractable epilepsy. So now, instead of removing the part of the brain, if I can make a device that you can insert in the brain and you can burn that area locally, right? Then it will save the normal region. So with that expectation and to understand the also efficacy of AEDs, like which AED will be more effective, right? And when you want to generate a new kind of drug, it requires billions of dollars. So instead of that, if you give an animal model in which a company can quickly understand the efficacy of epilepsy drugs, it would be interesting. So with that thing in mind, we wanted to develop this kind of probe that can go in rat's brain. Now there is a cortical column, all right, and cortical columns has lot of there is a cortex, and in cortex there are a lot of cortical columns. So cortex is on the brain, and then a lot of cortical columns which are close to each other. So if I insert a needle in a cortical column, there are different layers of cortical column, layer 1 to layer 6. Layer 4 is the input layer, layer 1 to 3 is a processing layer, layer 5 and 6 is the output layer. So if I have these electrodes placed into this particular cortical column, I will see how the signals are changing. This signals right from the brain is called electrocardiography. When we take the signals from the scalp, it is called electroencephalogram. So there's a difference. So now with that idea in mind, we fabricated this needle, and the commercial needles are $5,000 each. Each needle comes with a $5,000 price, and if some of the electrodes in this needle does not work, they charge it at $700, saying that it's a class B. Right, lot of expense for fabricating this, using it, or importing it. If we rely on the commercial companies. So we thought about fabricating our own microneedle, and we fabricated it. The cost of this microneedle is only $50 compared to $5,000. We can use it for several applications. We have used it for one, which I will show it to you today. And this needle is, you can see a coin. If you flip the coin, there's a Lincoln's photo. So this is just a representative diagram of our needle, how big the needle is. SEM image of a needle, you can see about 50 micron is a electrode size, 150 micron is the width of this particular needle, and we have inserted this needle into the rat's brain. So this is how the experiment was conducted. We use a micromanipulator. We inserted a needle. The needle was connected with a PCB, which was connected to a cyton board, and this PCB and the needle were wire bonded, and then it was inserted into the rat's brain. Rat was anesthetized. The animal ethical clearance for this particular experiment was taken from IISc Animal Ethics Committee. And this is an actual experiment where we have opened the brain of the rat, and this is a cortex in which we are inserting the needle. You can see needle is outside here, and in this case needle is completely inside. When you put the needle inside, you are able to see the signals generating from the needle. You can see here the baseline versus when you create an epilepsy. How to create epilepsy? If I load a bicoculant drug in this particular region, it will start creating a fit. So if you see that this is 1,000 microvolts y-axis, this is 100 microvolts y-axis, this is 40 microvolts y-axis. So you can see that there is a huge change in these seizures or the ECOG signal when there is epilepsy because of the presence of bicoculant. Now, if you load an anti-epileptic drug, if you administer it, then the baseline is regained, right? We are getting the baseline back. That means that the anti-epileptic drug is working. You create this is a normal, right? And then you have the epileptic activities, and then you are getting back normal. That means that after inserting the drug is effective to reclaim the baseline. That is what we have done as a part of this. This is a further understanding about 20 second signals, out of which we have taken 10 seconds to show you that there is nothing here. But when we go for epilepsy plus 1,000 by 1,000, there is a huge change. Again, baseline, there is no change. So there is a seizure when there is epilepsy. When we use AED, which is anti-epileptic drug, we can regain the baseline. Further power spectrum is done to understand that the filter that we have set is 100 hertz, and below above 100 hertz, we were not able to see anything, as you can see here, right, the frequency. We have also designed a microneedle with 3 shank, and we are performing experiments to use the 3 shank microneedles for several applications. These are the fabrication technique, 3 mass process. As you can see here, we have fabricated this needle. Again, we have kept next to 1 cent to understand the dimensions of this needle. We are in process of using this needle for several applications. There is another work on the similar domain, which is on flexible devices. So if you see, this is a group by John Rogers and team. John Rogers and team were the first one to understand, develop flexible devices with bioresorbability. What is bioresorbability? We have understood about biocompatibility, isn't it? But bioresorbability is when you implant the device and the device dissolves in the body, is bioresorbable. All right. So this is the flexible device implanted onto our head's brain. The signals are obtained, as you can see here. In this case, the flexible device was implanted. They created an epilepsy, and you can see a spike generated here. In this case, they created epilepsy by using penicillin and penicillin for 6 minutes. From base value, penicillin for 6 minutes shows a seizure value. So there are several applications. These are bioresorbable materials that one can use. This is all available in literature. That's why I'm a little bit fast on these slides. What are bioresorbable materials? Metal, semiconductor, insulator. And depending on that, we have also fabricated a chip that can be used as a bioresorbable material. We are in process of using it, but in the similar domain, we have created a flexible device. So this flexible device has a 32 electrode on the contact, and this 32 electrodes. So let me just go one slide back if I can show it to you. Yeah, in this 32 electrode, this and the tip of this device, there are 32 electrodes. Okay, this is a flexible device, and this device is these 32 electrodes are placed onto the rat's brain, as you can see here. Then it is covered and connected to a Saturn board. And let me show you an actual example. So yeah, here you can see very clearly. These are flexible device made up of PI, and the metal is platinum or gold. And these black dots that you can see are all conductive, remaining everything is insulating material. So we have used this flexible implantable device onto the rat's brain. So you can see here very clearly that we have placed the device on the rat's brain, and then we have sutured it, and then we were able to measure the change in the electrical activities. As you can see here, right, the left side, this is all baseline, because there are 32 electrodes. You can see 32 electrodes. The y-axis is 200 microvolts. In case of the epilepsics or of seizure, the y-axis is 3000 microvolts. These are seizures, right? And then again, in that case of when we administered the drug, the again we can regain the baseline. Again, you can see the y-axis is 100 microvolts. That means this device can be used to understand the creation of the epilepsy and to measure the efficacy of the anti-epileptic drugs. Baseline, seizure, baseline. Seizures are created because of the loading of the drug, which will create epilepsy, and baseline is regained because of administering of drug, which will remove this epilepsy. So it's called anti-epileptic drug. Right. So I think I have almost on time. It's 12:30, but I can take one more topic. Let me complete this one, and then I'll stop here. So this is on understanding the neonatal hearing screening. Now, you know what is neonate. Neonate is a baby born within a newborn baby. And the way to understand whether a baby can hear or not is to understand it's called the BERA system. It is used to understand the hearing capability of a newborn. Now, this system, which is called BERA, cost about 14 lakh rupees. So most of the secondary clinics, most of the primary health centers, they don't have the BERA system, but the secondary clinics and tertiary clinics would have. That means that a lot of babies born in our villages, right, they will not go through this particular screening technique. That means that when they grow 2-3 years old, at that time their parents would know that they are not able to hear. So the cognitive ability of a person will degrade when you are doing this at a later stage in life. And that's why it's very important to develop a tool that can be used for a neonates in the primary health centers where this system cannot be used. In this case, the affordability is very important. What we have thought about is that we will develop this tool for neonatal hearing screening, and the commercially available tools are shown here. Like I said, is cost very high, and they give a signal and like an audio signal, which we call evo potentials or ERPs, and then they measure the EEG coming from the frontal region. That EEG coming from the frontal region will tell whether the baby can hear or not. Now, what we are right now doing is only looking at the half circuit or half hearing cycle. To understand the full cognitive ability of a brain, you need to understand the full cycle that comes when you give the ABR signal, which is called auditory brain response. So there are OE and ABR. OE is autoacoustic emission, and ABR, as I told you, is auditory brain stem response. So we were proposing of understanding the complete cognitive ability of a brain, not only the hearing capability, and that's why we proposed two different biomarkers along with OAE and ABR, which is mismatch negativity and the ERP. What is mismatch negativity? Suppose a mom says da 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 da, right? So when there is a change in this, that is called mismatch negativity. So when this happens, there is a change in the signal in the EEG. So we were planning to develop this particular band, and now we have developed. At the time we were planning, now we have developed. We have got ethical clearance from Hubli Kim's Institute to use 200 subjects, newborn neonates, and place this band on the neonate's head, and to understand the signals so that this band itself, the complete band, is costing only 40,000 rupees compared to 14 lakh, right? So there is a region. This is a band on the young adult to understand all this EEG signal that comes into play. We have used the MATLAB for based ERP extraction mechanism to remove the artifacts and to create the actual things. You can see the band here in the video. So there is a pillow on the back side, so it will not cause any kind of deformity or pain to a neonate. And there is a sponge also in the band itself. So this is a fabricated one that we wanted to test with young adults first. There is a space for the EEG electrode. This EEG electrode we have bought or we have imported from Florida Tech. You can change the size, right? Because different dimensions of the head. So then the comparison with the existing product, and we can do brainstem, cortical, head stem system. Portability is very high, cost is very low, ease of use is very high because this can be used even by the semi-skilled personals like ASHA worker. We are already at TRL 5 level, and now we are going to do these studies to understand. Our idea was to make a non-invasive, objective, multimodal approach to understand neonatal auditory pathways. It can be deployable in primary healthcare centers. The existing possibility of auditory threshold identification and neonatal epilepsy screening system development by increasing number of electrodes. It is available. We were trying to place this band in this kind of form so that for the parents, it not be that you are testing the baby with some electronics, right? You just have a toy. Of course, they should know that it is for understanding the neonatal hearing screening. But we were thinking about baby friendliness. These all things will come once we have enough data with us. So the market size is really high. The 4 in every 1,000 children have a hearing loss problem. Over 1 lakh Indian babies are born every year with a hearing loss deficiency. These statistics are twice compared to developed countries, and that's why the competitive landscape and in next 5 years, what we can see, right? We have mentioned in this particular slide some results from the mismatch negativity. We were able to see the signals very clearly. We were also able to see the signals signals from P300. P300 is a signal that comes randomly, which is nothing to do with the original signals. I'll give an example of P300. These are certificates from IISc, Indira Gandhi Institute of Child Health, and Queen's Hubli to use our system and try it on neonates, babies, healthy babies born within 1 to 3 days. The commercial systems are available in the same hospital. They will first test the babies with commercial system, then we will put our system and we will see how accurate our systems are compared to the commercial system. So here I'll stop. There are many more slides that I can present. In fact, another 2, 3, 4 hours we can talk, but respecting the time of the participants as well as committee members, I'll stop here. The point about P300 is that, let's say there is visual P300 and there is a audio P300. The visual P300, I'll give you an example so you will easily understand. Suppose you see every time balls, right? Blue ball, blue ball, blue ball, yellow ball, red ball, and suddenly there is a checker box. The checker box, you understand, like a chess. So this is not what you are expecting. In real life scenario, suppose I'm sitting in this office, right? And suddenly I see that there is a line in the office, which line is not expected. So suddenly there is a P300 signal, which is positive peak at 300 millisecond that generates from our brain. To do that, we need to have 3 electrodes. To measure the auditory evoked potential, 1 EEG electrode is enough. So what happens is you give the signals through the ear and you measure the EEG from here. Now, why there is a EEG and ECOG? EEG is electroencephalogram, ECOG is electrocardiography. Electrocardiography is when you take the signals from the brain, electroencephalogram signals from the scalp, and you have seen 1020 system, sometimes one single electrode system, sometimes. The difference is that because of the impedance provided by the skull and the tissue, from the brain all the way to the scalp, there is a reduction in the overall signal. If you know the ECGs of few millivolts, EEG of few microvolts, that's how the reduction generally happens. So anyway, I'll stop here. I'll take any questions from the participants. If not, then we will finish the lecture here. Thank you very much.
Participants, any questions from your side?
Hello. Can you hear me? Hello.
Yes,
Yeah,
yes,
hello.
Yeah, you are audible. Please go ahead.
Yeah, hello, Dr. Pandya. Thank you very much for the insightful talk, and it was very informative presentation about your research work, ongoing research work. I am Ganesh Bapat. I am faculty in the Department of Mechanical Engineering at BITS Pilani, Goa, and I work in biomechanics and assistive devices. So I have a couple of questions for you, and my questions are more of related to research methodology and doing clinical research in India actually. So in India, especially, we find that engineering and medical institutions work in isolation. And in such scenario, how did you come up with such strong network of clinical collaborators and get those very detailed and clear clinical problems? So is it like chicken and egg story, like first collaboration and then you got the problems, or you had the problems and you approached collaborators, and how did you build that network?
Right. So I had some earlier work done from my PhD postdoctoral work on which we worked on certain problems. At Harvard Medical School, I was working on understanding drug efficacy for chemotherapy, cancer, working on AST, which is antibiotic susceptibility studies. In Maryland, I was working on breast cancer project. So I had some preliminary data. Now, after I joined here, I got to know few doctors. I approached them that I'm interested in working on this. And once they started working on with me, they found that it is a interesting collaboration, fruitful collaboration. There is a frequency at which our frequency are matching. So it was more like a resonance. And that's why they introduced me to other doctors. So it was kind of starting with one group, and then a few doctors kept on adding. So that's how it happened. I agree with you that generally we work on isolation, but you can try connecting with some of the doctors right in Goa, right? For example, Sunil Band is there, right? So he's trying because of his understanding about biochemistry for healthcare application, right? So I used to work with him when I was in IIT Delhi on a project on aflatoxin and biosensor of aflatoxin. Then we fabricated biosensors for his laboratory. So you can start depending on what kind of research area you are working on, and try to contact few people. First, try to contact within the city. It's always good to have someone in the city rather than other places, because very difficult to keep on long-term collaboration. So that is my first opinion regarding the question that you have asked. It's more like you should have some kind of data. Like even Tagesh asked that, okay, if you want to go for getting the blood, what should be that? So I said, okay, get data from the PBS. And once you have that, people will have more confidence in your research. Sometimes it's more like an institute also. Like if you represent institute like IISc, NIT, IITs, even BITS, people will wants to start collaboration. But I personally believe that until I have some data with me, it's easy to talk rather than implement. So if you have implemented, people will have more confidence in your work.
Correct. Yeah, right. And one question follow up to this question, again, general question, like doing this ethics approvals, biosafety, and it takes a lot of time and a lot of revisions as well, because I did few during my postdoc and PhD as well, and gone through that procedure. And I'm really fascinated that many approvals for different studies, everything. Of course, it is required, but then what are the challenges in doing such clinical or medical related studies, and how do you overcome those in your research, and how do you manage that actually?
Yeah, but maybe because I am in IISc, I will say like that. I faced minimum resistance from the Institute Ethical Committee to approve my ethical documents. Of course, there were revisions, but it was very fast. Ethical committee does not look too much into the technicality of the work. They look more from the what is the number of samples that you are going to use, how much it is affecting a patient. If it is not affecting a patient, already a patient is coming to a hospital and he's giving blood, for example, then can you ask the patient and get the consent to take 2 ml of blood from the so much of blood that somebody has given? Then it's very easy because you're not facing any other problem, right? But if you're proposing a new project where only exclusively for your technology patient has to work, then it will take a bit of time to convince the committee. So what I would do is, like for example, I wanted to start with a project on eNose to screen some lung cancer and the liver cirrhosis patients. So I asked the committee to give me approval for generating the normative data. Normative data means data from the healthy subjects. And those healthy subjects were subjects from my institute. Once I had that, in the next cycle, I said that, look, I have got the data. It is interesting. My sensor is working. If you can get approve the ethical committee for using it on these subjects, I just want their breadth. And I want to compare with this normative data to see how much difference is there. Then they immediately agreed that, yeah, it is very interesting because you can see some signature. So sometimes to put it a little bit way that is easier for the committee to understand, I think that would be a right approach. Since you already have experience of going through multiple committees, you know that the committee consists not only of scientists and engineers, but also of and then societal representatives and other people, right? So
Yes.
You need to like put the things in very easier way to, and then connect with how it is going to be useful for the overall society. Then I think it will work. But I personally never felt any difficulty. Yes, the for getting the IISc approval, I had to first get approval from the hospitals. There I had to go there and present the work, and there I faced a lot of questions. But if your research is clear, you have the answers for most of the questions, I think the committee would approve it. In India, it is way easier, comparatively easier than in US. It was very difficult there.
Right. Yeah, I have experienced both actually.
Yeah.
So I just asked like how much time it takes. But yeah, anyways, thank you very much for elaborating on this and sharing some of your experiences. And thanks for the wonderful talk again.
Thank you, guys.
Any further questions from the participants? No. If there are no further questions, thank you very much, sir, for such an interactive session. Even it's very difficult to find a question for me also for this presentation because it's a very new topic for me. But I hope like as I have found it interesting, other participants also would have found it very interesting and very enthusiastic presentations.
Thank you.
See, definitely we all will be in touch with you, and any case, let's be in contact, sir, for further any studies.
Sure.
Thank you very
sure.
much, sir, for giving us the opportunity to interact with you, and thank one and all for again cooperating with us.
Yeah. Thank you for having
you
me.
very much, sir.
Bye for now.
Yeah, yeah.
Bye.
Thank you very much, sir. Bye-bye. Okay, participants. So I think that's the first session of the FDP. Now it's closed now, and thank you.